Okay, so why is microscopy so important? Well, we now have the human genome. We can go and sequence something really fast now. We can go sort of the min ions, okay, and go down that type of route and do our single cell genomic analysis. It will tell us if the RNA has been transcribed. But fundamentally, it doesn't tell you if the protein is being transcribed. So as good as genomics is, fundamentally, it's useless. But don't fear, because now we can isolate that cell or cells, we can purify the proteins, and we can take it to the proteomic lab. Then they can tell us if the protein really exists. Fantastic, it's really there. But it doesn't tell me where it is. It doesn't tell me if it's functional. So proteomics is also, I would argue, useless. Now, of course, as a biochemist, we can do biochemistry, biochemical analysis. I can purify that protein. I can now put it into a cuvette. I can put it into a spectrophotometer, a fluorimeter, and I can now see the enzymatic activities. I can put it into my SPR, look at my binding affinities. But it's in a cuvette, not in a real cell, not with all the cofactors and the environments that are so important for it to function properly. So even biochemistry is useless. You're catching on. Which is why microscopy is the #1 technology that all biologists should be using. Okay. It's true, huh? Because now I can see that my protein is really there. I can see it is localized to the correct place. I can do biophysical interactions with it. I can do my fraps, my flips, my flims, my flaps. Okay? I can do all these 4-letter acronyms. If you can think of another 4-letter F word, don't use it in this audience. Okay. And of course, I run my imaging and cytometry lab, and we have to have the significance of looking at many data sets. Okay, so we have to think about measurement and quantification. Okay, so hopefully that's got you engaged. I was really thinking about making light work and make light work more for us, getting more information from the same data sets. So this is really going from 2D imaging through to 3D live cell imaging, through to super resolution, understanding and seeing more detail of what we were looking at to start with, so we can see really where our protein is and really what it's doing. And then correlating it to look at how it maps onto the ultrastructure of the cells. And that's why you're all here today. But really, we fundamentally, as a microscopist, our responsibility is to gain greater information from the same sample, so we can understand it in more detail. Okay. So, as I say, we have the service lab and we have the research lab. This talk is going to have to go through fairly fast. We'll skip those. But the lab has several areas that it's been involved with in some capacity in its development. Through from label-free imaging, so tychography, through to increasing the sensitivity to minimize the amount of perturbation on the cells, so the area scan, through to multiphoton, light sheet microscopy. We've had a hand in a lot of these different technologies, right through to a different technique, which I'll introduce later, and correlated imaging with Lucy, which I'll also show a little bit of. So I'm going to start with a label-free imaging. So I'm going to take us back a resolution and think about the cells to start with. So really what I'm trying to do is not add any labels to it, because as soon as if I take a primary cell line, if I extract it from the rat spray, okay, I don't do that personally. Okay? But if these cells are, if these are prostate cancer cells coming out of a human biopsy, okay, let's not go into too much detail, otherwise a lot of men will cringe at the thought of it. But you can't then go and genetically modify these. You can't go and add CFSE into these because you will perturb that cell. You want to minimize that perturbation. So label-free imaging is really important to start to understand what's going on. So what we want to do is convert a classic light microscope image, which I'm sure some of you will recognize, unless some of you are hardcore electron microscopists and just think, what's that? Okay? But what we want to convert it into is something like this. A high contrast, which is easier to segment, easier to quantify. And this is a label-free image. It is not fluorescently tagged. It's not phase contrast, it's not DIC, it's not dark field, it's tychography, a different technique. Very, very briefly at lightning speed, we pass coherent light into the specimen, and the light passes through the specimen straight through to the CCD camera, and the CCD camera images the diffracted light pattern whilst it's looking directly at the laser beam. So we use far red light, the camera has to be looking directly at the laser beam, so the laser beam has to be significantly attenuated, turned right down, otherwise it will blind the camera. So the phototoxicity is very, very low. We then take a series of overlapping XY tiled images where the diffracted light pattern changes. And from that, we can take those series of tychographic diffracted light patterns, put it through the tychographic iterative engine, through the Fourier transform, which will then give us at the back of that a phase, a sort of bright field type image, and a phase shifted image. What that enables us to do, enables a computer to do, is to segment it much easier. Something we've been struggling with all week. We talk about 3D imaging and trying to improve contrast to give us better segmentation. And we had the same problem with bright field microscopy. Actually segmenting in bright field or DIC or phase contrast is very difficult. But now we have a positive discriminator. We can segment even in high fields of confluency. But importantly, we can do live cell imaging as well. So to start off with, watch this cell. Okay, when you should see the chromosomes align and pull apart. But then also watch this cell. I do like this. That's probably one of my favorite cells in this movie. So these cells aligned, chromosomes align, divide. These cells are just divided. One sister goes this way, the other sister goes that way, and goes, no, come back, and chases after its sister. Huh? Poor thing. Okay. Now, the intensity of those cells is quantitative. The brighter the cell, the bigger the volume, the bigger the mass of the cell, which means we can now look at mitosis, we can look at G0, we can look at G1. We don't quite know where G2 is, hence it's a gray area, back through to mitosis again. So without labeling it, I can look at the cell through cell cycle without that perturbation. Now, this led us on to looking at primary hippocampal cells. And I won't talk about Hannibal, which is a cell that contaminated our previous ones, but I'd like to introduce you to another cell. So, first of all, we have a cell that's just died here. We can tell because the phase shift change has a different trend compared to cell cycle. Protein's diffused, this cell comes down, eats it. See this apoptotic cell just here? Wait for it, it's going to eat it. Go on, go on, go on, go on, go on. Oh, fat boy. Okay, I'm going to take you back. I need to work out how to rewind this on this now. Okay, I'd like you to watch this cell just here. This cell does nothing. It just sits there next to an air bubble, just sits there. It gets bigger and bigger and bigger until it's double the intensity, and then eventually, get on with it, it divides. So we've got two identical cells. Now this cell on the left suddenly gets hungry, and it goes off eating everything, okay? Its sister, huh? I can't be tossed. I just can't be bothered. He's just going to stay there. This one is still eating everything. There's a lot of food up here. Watch this. I'm going for that. Huh? But this cell here has done nothing until it divides again. Go on, go on, go on. Yay, no. And then one of them eats the air bubble. It goes out the focal plane, comes back with a huge amount of indigestion. Wait for it. Go. Okay. Now, the first cell that divided and got really hungry, we called Dyson. Now, I don't know how many of you know what Dyson is. It's a kind of a vacuum cleaner, okay? But I mentioned Dyson for a very good reason. Firstly, it is an example of British engineering. Huh? So, Tom, those from America, those from Germany, we too can do engineering. We don't overcomplicate it. It's elegant and yet simple. It's efficient and quiet. And it's a world leader. Hey! But it is still a vacuum cleaner. Okay. So that cell that went off and got really hungry, we called Dyson. Okay, because it just hoovered up, it vacuumed up everything. From the same movie, we've just digitally zoomed into one area. And you'll know, hey, come on. You'll now see Dyson come in. And this is sort of fidelity we're getting off this. So you can see the bits that it eats. You'll see these bits internalize into it. You can see it dissipate and dissolve out. Look at the debris on this new right here. You'll see the debris just here comes up, cleans it off, disappears off. However, the new rights down here, look, these will actually undergo division. Okay? Now, all of these movies, from the start to the end, this is just a sort of snip, was around a 6-day time lapse of continuous imaging. Okay, so it's live cell, it's non-invasive. But that got us thinking, these two cells were totally different. Ah, that's cool. So let's go and look at these and study these in more detail. And the nice thing about this is we can study these even in large fields of view. That movie didn't work. Let's try this. So now we could take huge fields of view, 3.2 millimeters, and we can get all this information at the same time. So the significance now, looking at these different cell types, we can then isolate them, take them for further analysis downstream. But now we know more about their behaviors. And we can do cell tracking, so you can track the cells. And this led on not from Dyson, but to Usain Bolt. Okay, so Usain Bolt, we thought, I know how to win this game. Okay, so he is also a world leader. Okay, but in this race, he used drugs. Now, I'm not insinuating Usain Bolt uses drugs. Okay, but in this case, we drugged him to make him slower. Okay, so this should mean we can beat him. Yay! By the way, that's a great way to dope, isn't it? Huh? Don't dope yourself, dope the opposition to make them slower. Huh? It's so much better. They then get banned if they win anyway. That's a fantastic idea. Okay, so the idea is to slow them down. Okay, they compete against a control sample, which should win, but guess who wins the race? Yeah, Usain Bolt wins again. Okay, so we did loads of cells. We followed their individual tracks over time. And so these are the drug-treated. You can see that the ones that are drug-treated are all slower on average. But the fastest cell we had was Usain Bolt, the fastest by some way. Okay. Now, most times we just go, that's an outlier. Maybe it is just an outlier. Okay. But there's a trend here. He's also really fast. And if you think about your different drug treatments, think about your chemotherapies, for example. 99% of the cells will conform to this, but your 1% will not. And it's that 1% that will kill you. Thanks, Usain. I've now called him a murderer. Huh? Okay? So we have to think about this. We're getting all this different information from this. So not only can we segment it, we can track them, we can get all these sort of phenotypic, new, different phenotypic markers. We can look at the mass of the cell. Was Usain bigger, smaller, fatter, slimmer, longer? Okay, was his volume big? Was his greater spherosity? Did he have more granularity? We can get all these different markers that we don't usually look at, which is great. But fundamentally, once we've tracked him, it doesn't tell us where our protein is, does it? So we still need to fluorescently tag it. So, of course, you can correlate label-free with fluorescence microscopy. You can go from your phase image through to your fluorescence image. So you can still do your normal fluorescence microscopy. And, of course, you can do multiple colors, so forth. But that's useful. But still, we need to see more. And as soon as I add those labels, I risk perturbing my system. I want to minimize the amount of light on my specimen. Okay? And actually, we were fortunate that we were a beta tester for the LSM880 and their ARIE scan. And this kind of enabled a lot of users to get super res with improved sensitivity, okay, which is kind of critical to my user base who are, I've got to be careful, it's being recorded. I was going to say something else. That just want an easy microscope to use. You can read that as you want, okay? So the ARIE scan, a very brief introduction. Okay, so instead of using the normal scan light path, it takes us through to this different detector that's just here, which gives us this array of pinholes, which improves both the sensitivity and the resolution of the microscope. And critically, it's a confocal microscope, which means we can now use this in 3D. We can use it deeper into specimens, any confocal depth. Remember, if we go into a root tip, if we go halfway through it, you're not going to see what's going on in the middle. Whereas now it's a confocal, we can see that at much better resolution. We can then form our three-dimensional rotational images, which are as good as any SEM image. Why use an SEM? Can we use my confocal? Okay. But seriously, what sort of studies can you look at it? So you've got to think about sensitive issues. If you're time-lapsing, you want to minimize that light so you don't cause phototoxicity. And the nice thing about the detector, the gas detector is kind of up there, is one of the more sensitive detectors in confocal microscopes. And this, you should do a scan of 1. If you're having a scan of 4, you improve the signal to noise. But with just one scan here, using the ARIE scan, it's very similar in signal to noise, if not slightly better. So now I decrease my laser power 10 times, average of 4 with the ARIE scan. So now at 10 times lower energy, I'm still getting much better data sets. Decrease it to 50 times less laser power, I'm now not really getting useful data. I can still get useful data out of this. It's about twice as sensitive compared to the normal internal detectors, which means I can use half the light. Now, actually, I'm sorry. I know ZEISS have kind of sponsored a lot of this meeting, but I couldn't resist this. They'll kill me at the end of this. This is one of their slides. They didn't know I had this. Okay? No, I've not hidden. I've got this slide off. We'll just ignore that bit. Okay, so they've kind of shown how the sensitivity of confocal microscopes have got better and better and better. And they argue they're nearly halfway to the sensitivity of where we can really get to ultimately. Now, if this detector is twice as sensitive, what they're saying is, look, we are nearly perfect. Huh? But it's not. All they've done is increase the bar. Okay? So they're still only halfway there. Okay? It's just that halfway is now much further, is a much bigger target that we're aiming at. I couldn't resist that. Okay. So the detector array enables us to improve both the sensitivity and the resolution of the microscope. And so some of the very first images, the first sample we put onto it was this one. And it enabled us to look at these mitotic cells, and we could then see the resolution improvement between the two. But it's not just in XY. Remember, it's also a confocal microscope, and it also improves the resolution in Z. So we could then Z stack through this and see the improvement in X, Y, and Z. So this takes us to around 300, 350 nanometers in Z resolution, approximately, very roughly. Still not quite 50 nanometers, which Lucy was getting with her serial sections for the fluorescence images. I only said that to make sure Lucy was listening. Huh? What did he say? Now, to prove the resolution, Z is now on the x-axis. If you can't get your head around the Z being on the x-axis, just turn your head sideways. Then it works. Huh? Okay, so you can improve the Z resolution. Is also improving in the Z domain as well as the X and Y. And it's a confocal, so I can get that resolution even in tissue samples. So in this case, this was one of Mark Cole's samples. Okay, so we use a tissue section, and we can Z stack through the tissue section, looking at, actually it was a lymph node in this case to be precise. And we can do the area scanning with the same sensitivity, the same resolution as we go deeper. We can look at bacterial samples. So here's our 3 just here, the same 3 as here, 2 and 2. Zoom in on these, and we can now see, we're starting to see structures and details that we couldn't easily see before. So if we now want to correlate this, we're getting a better idea where the light microscopy is, where the fluorescent staining really is. It's not just a fuzzball of fluorescence. Okay, and you can do it in multiple colors. It's just a normal sample. No sample prep, just put it on, get your image. Which is why it's so nice. Okay, that's why my user base uses it. Actually, this is one for Homer Simpson, because it's donuts. We've got a guy called Sean Sweeney in York. And we have these open days at York. And in the atrium, when we're trying to encourage the students to come to York, there's always donuts on the side. Huh? I don't know if they think, oh, we'll go to York, there's always free donuts. Surely they're not that gullible. Maybe they are. But Sean always goes in and goes, Pete, do you want a donut? What was that? Sorry, Sean? Do you want a donut? I can't resist a donut. Yeah, Pete, have a donut. I don't want a donut, Sean. Why are you so crazy? Every time. I don't know why I'm always in the atrium with Sean, but do you want a donut? By the way, it's my best Sean Connery impression. Huh? Okay, he's also Scottish. It's the only Scottish accent I do, so if Sean's not quite like that, it doesn't matter. And I never understood why he was after donuts until we got the area scan. Because this is what his neuromuscular junctions look like. Obviously not his own neuromuscular junctions, that would be kind of perverse, okay? From the samples of his Drosophila and stuff that he's looking at. And then we took this image, and we could start to see ring donuts. If we zoom in on this. Okay, so what he was always looking for was ring donuts. So I don't know if he ever knew that. Okay, so that's why he's compelled towards ring donuts. I can't wait. Now he's going to want to do 2-color, isn't he, yeah? He's going to put red in the middle of it, so now we're going to have to get jam donuts. But of course, as we improve the sensitivity, as we improve the resolution, we have to change our sampling rate to fit Nyquist. As we fit Nyquist, we're now going to have to increase the file size, the Z dimensions, the XY dimensions are all increasing, our temporal resolution will increase because we can speed it up, and our file sizes are getting bigger and bigger. So I'd actually like to introduce the LSM880, which is an example of ZEISS engineering. I bet they don't know what's coming next. Okay. Elegant and yet simple. Efficient and quiet. Thinking low noise here. Signal to noise. Quiet. It's a world leader. I'll pass you a compliment. But it is still just a confocal microscope, guys. Okay. And if we want to do live cell imaging, if we want to do in vivo imaging, we have to think beyond the confocal microscope. So then we can go on to multiphoton microscopy, which enables us to use far red light to go deeper and deeper. So this is a biofilm, and we can go deeper and deeper. The single photon dies out. Deeper, the internal detectors die out. And deeper and deeper and deeper, which gives us a much better idea of what's going on, greater depth, greater information. And then we can put that, there's a bit of chromatic aberration in here, so don't kill me, okay? But it gives you an idea. You can see this biofilm has valleys on the surface. It's not just a nice biofilm that's thick. We get an idea of what's really happening in the 3 dimensions with this biofilm. We can then take those 3D images, and we can do it over time. So here we have a vascular system, and we can watch the cells go through. Okay, so we're just watching the live cells come in. We've got another live cell that should come in here at any moment. Here he comes. Through. We have another one that comes in. This one doesn't like it, comes back, chases it away. Goes, I'm not having that. It chases it back. Okay, we'll give up. Go. Right, so cells have personalities. They are all different. Okay. This I like this cell. Huh? This one's in love. Watch it. Huh? Comes down, gives this one a little kiss. Goes off. Huh? He really likes to interact with this cell. So even at that depth, we're getting ideas of interactions. We're sampling it. So that's a cell that's interacting. We want to capture at that moment in time. But the problem is with multiphoton, you can only image for so long. We are putting effect as you go deeper and deeper, it still becomes phototoxic. So we need to increase the sensitivity and the temporal resolution. And this is where light sheet microscopy comes in. Okay, so it really helps us in vivo and for larger sample types. So we've got an open spin system, and we also have the ZEISS system. And so we can now do our Drosophila heads. So back to Sean. You can go right through the Drosophila head really fast. So now we're collecting data much more rapidly than we were with a confocal microscope. So we go right through to the sort of sensory glands at the top. Put that in 3D. Gives you an idea of what that actually really looks like. Actually, it's only its sensory glands at the end of its snout that are GFP tagged. The rest of it is autofluorescent. Okay? So even Drosophila's bright. We could do spheroids, and for the 3D, because you can rotate the sample around and see it all around it, you can then see asymmetric cell patterns that you wouldn't have necessarily seen if you just looked from one aspect. We could go on to, I'll flick that one, into your neuronal systems. So this is a zebrafish in this case. And of course, these are again bigger areas, more sampling, stitching them together. And then you want to do it over time in multiple colors. We can watch, actually, there's no vascular system just here. There's no blood vessels. But as it grows and grows and grows, suddenly the vessel will form, and you'll start to see blood pumping through here in a minute, just at the end of the movie. It will start pumping. It will start. Come on. Start. Start. Now, okay, you see the blood pumping through. So we can watch this development stage. And then you know that's a time point maybe that you want to go and capture that and take it down for your other microscopy techniques. But of course, this gives us a problem, a brief history of file size. And we talked about this yesterday in the open forum discussion. Because we're increasing all the time our temporal resolution, the number of dimensions we're looking at, okay, the resolution, the volume that we want to collect, which of course costs more. So we can go from our movies to our sort of root tips here, 300 megabytes, to our biofilms, which comes down to around 1.95 gigabytes as we collect it. And of course, that limits out for the amount of time we can image, which takes us on to this, which carries on imaging, bigger frame size, number of angles, number of stitched together, and we're getting up to the terabytes. And we heard earlier in the week that we were talking way beyond terabytes, but this is realistically what we can actually capture relatively easily within the lab. I used to love these days. Huh? I used to be able to get 4 of these onto one floppy disk. And I used to have hundreds of floppy disks. I still have them. I can't use them. I still have them. And of course, that cost, just that one data set cost me €300 just to store, just for that one data set. So there's a cost. So all this comes with a cost itself. And really, this is just a start. Our data sets are getting larger and larger and larger. Ultimately, though, for all those samples, and one of the reasons for this meeting, is we want to go into even finer detail, finer resolution. And this really takes us to looking at what we can resolve with a light microscope. And we know we can't see below the diffraction barrier. We can with super rays. We can kind of get there through different means, but it's difficult. Okay? We can't really see if these green, the red, we've got co-localization, the yellow here. We can't really prove that's co-localized. Our resolution isn't good enough to show what's really happening on a protein function. This is on a membrane. Functional units on a membrane. Okay, do we have 1, 2? We have dimers. Do we have tetramers? What's making that pull? What other proteins are coming into that functional unit on a membrane? We still can't readily do that, even with super res microscopy. Okay? And most of super-res techniques, they can do multiple colors, but there is a limit, and you start to deteriorate your resolution when you're cross-comparing the two. The sample, we've heard, the sample requirements are quite restrictive in many cases. And the likelihood of seeing every fluorochrome is very low. Okay, if you're using an antibody for palm, the chance that every antibody has a fluorescent tag on it that's viable, not that high. Your GFPs, have they all matured? Don't forget, you've got a maturation rate of GFPs. They won't all be mature. They won't all be fluorescent. So you will not see all your proteins when you're using different technologies. Okay, so it's very dependent. So if you want to look at functional, it's great for protein patterning. But to look at functional complexes, it isn't so easy. So this kind of got us thinking, and we wanted to change that. We wanted to try and improve that side of it. So this is the collaboration we have. Lucy talked briefly about this yesterday morning. Was it on? No, 2 days ago, or whenever it was. It's all a haze. Yesterday. So we got with Lucy, some chemists at Brunel, Joel, Catan, Delmic, and our funding bodies down here very generously sponsored and supported this with great enthusiasm. And we wanted to bring and develop the best of light microscopy and the best of electron microscopy, because we can take both advantages and bring them together. Then we want a win-win. Okay. So my part of this is trying to bring the EM into the LM world. I want to use an electron microscope in a light microscope sample. Okay. Lucy's side of this is actually taking the LM and putting that into the EM world and getting light microscope images of the EM sample, with the improvements of the Z resolution, with the ability to do super resolution to map it onto the fine ultrastructure. That's Lucy's part of this, which she talked about yesterday so brilliantly. So in our case, to start with, conceptually, we were making new probes. And with those probes, we tested them with a Catan monocle system. And we're using the electron beam just as we would use a confocal microscope beam. Okay? So the confocal microscope beam must hit your fluorescent dye for it to emit. Now, in our case, we're developing probes that are electron beam excited. So when the electron beam hits it and it emits, it can only have come from where the E-beam was. So it's limited by the electron microscope, not the emitted wavelength. What does that mean? Well, we had to get new chemistry for this. So we went after different phosphors, which are both excitable in the light microscope and also in the electron microscope. And different probes have different emission bands. So we can do multicolor experiments with these. And they have very narrow emission bands, which means that the chance of bleed through is minimized. It's not perfect, but it's minimized. And this is in the SEM. These are the probes in a vacuum, in an SEM on a silica chip. And this is at the resolution of a confocal microscope. So these are 100 nanometer pixels. So roughly at Nyquist for a confocal microscope. Now we take it with the SEM and we improve the resolution, we magnify it, and they're the probes that we can really see that's there. So that's the difference for a light microscopist, which is where I'm coming at with this, from here to looking at this, just by increasing my magnification. And because we're using phosphors, all the phosphor probes are emitting light. So now we're seeing all of them. They're not bleaching. They're very, very photosensitive. And put them onto a membrane, so literally just scattering them onto a membrane of a cell, and we can now start seeing all these molecules. We've got some autofluorescence behind. But again, a narrow pass filter, we then clean that up and improve the signal to noise. However, the probes themselves are around 13, 14 nanometers in diameter, so it's not perfect. Okay? And actually, if you think about the size of an antibody is around 10 nanometers in dimension. So for a light microscopist, going much further down in resolution doesn't make a lot of sense, because the things we're having to tag it, the things we're labeling them, are as big as we're limiting to our resolution. But what we really wanted to do is bring this into a light microscope. As a light microscopist, it's very easy for cell biologists to take their cells, grow them in a Petri dish, fix them, label them with an antibody that's stained, and image them. Okay, that's probably the most common uses we have on the microscopes. So we actually looked at the Joel and Claire scope. So we have an inverted scanning electron microscope and a light microscope above this, and a Petri dish that sits between the two. And so we have the E-beam underneath it. We have a thin silicon nitride membrane here, which enables a sample to remain in buffer. So it's just a normal, for biologists, it's a specialized Petri dish, but to them it's just a Petri dish. Okay, it's quite simple. And then they can image it with the light microscope, but also image it with the electron beam. And if we use that like a confocal microscope just to scan, we should be able to get better resolution for our protein patterning. So yes, these were these images. This is kind of great optical microscopy, green, red, and you can zoom in on this and see your higher resolution details. And it gives you super resolution microscope type images. So if we take the tubulin, and we can take a stain one here, but this is using silver enhanced, which isn't always the easiest thing to execute for Joe Blogg's biologist, a non-specialist. But you do get very high resolution detail. What we really wanted to do is make it easy at normal common practice for biologists. So that's what we've done. We've taken in to proof of principle is to take the antibody to GFP and then have an immunogol tag on it. So it's just a straight immunolabel, PFA fix, antibody label, and image. And so we can take our GFP tag cells, ones without GFP, so we have our control specimens, and then you can see the high resolution, high contrast, electron excited super resolution microscope image. And you can, that's pretty impressive, huh? Okay, let's zoom in on it. And then you can start to see all the individual proteins that are there. Okay, with little worry about it. We can look over here, and then we can see the protein patterning and the structures of where those antibodies are actually tagged to. So the next challenge is to take the probes that were cathodoluminescent and put them onto the Clairscope, and then use the electron beam to excite those to give us that 20 nanometer resolution back at the detector. So this isn't an electron microscope in the true sense. Okay? This, to a light microscopist, is a super resolution microscope. But a very simple application. All you need is that antibody to get your data. And it's really looking at functional units on membranes. That's really where we're aiming it at this moment in time. But as I say, it's a collaboration, and a collaboration with Lucy, and actually it goes the other way too, because these probes will also be useful for exciting inside the electron microscope. Okay, we can correlate it, we can use them as fiducial markers, we can use them to tag the cells, or looking at the proteins within the cells, and because they're very photostable, we can also image them for a high length of time. And actually this is using the GFPs you saw yesterday, with the idea, but the other side of this is to be able to take your LM images inside the electron microscope itself. And Lucy had better pictures yesterday, which she hasn't given me. Huh? I also noticed she got a new logo, so I was desperately searching on Google yesterday to get a new logo and putting it all, copy, paste, everywhere through it. Okay. So it's kind of addressing both sides. Okay. We're looking at taking the EM into the LM and the LM into the EM. And I think we just need to make it easier for the biologist to use. And that's really critical. Just make their lives easy. Means we have to do really hard work and complicated stuff to get there. But until we get there, until we create the problems, we'll never have the solution. Okay, so we are creating problems to find those solutions, which will help the biology. And that's really what we're after. We're not after developing a new technology to get pretty pictures. We're here to address and get better science. We're really looking to get better biology. So we have to think wider than ourselves. So I introduced at the start, the imaging and cytometry, microscopy is everything. But now as director of all these labs, I have to think a bit more holistically. Okay, I think I have to think a bit bigger. So we talked about why microscopy is better than genomics, proteomics, biochemistry, and I am right. Truly, I am. But if we think about correlated microscopy, we can go through to our live cell, tychographic, label-free imaging, minimal perturbation, getting these new phenotypic markers of our cells. We can then take that quantitative information, then label the cells, and we can then localize the protein where it is. But we have an idea if that was a fast cell, or a slow cell, a heavy cell, or a light cell, a long. We've got all these different characteristics of that cell that we can map it onto. So we're getting more. I bet not many of you do flow cytometry. If you give a blood sample to a doctor at the hospital, he will use 9, 10, 11 different antibodies to phenotype your cells. Now we're looking here at phenotyping our cells without having to go through all those labels to understand it better to start with, to pick out the rarer cells or the common cells, if they're really reflective of the ground population or not. We can then take those to do our super resolution microscopy images, knowing all this information. And then when we know this, we can take it through and really get that fine ultrastructure. So we're mining down into greater and greater detail. But we need to think bigger than that. Correlated microscopy is not just correlated microscopy with other microscopies. We should also be thinking about matching our data and getting our information, then matching it with genomic data, proteomic data, metabolomic data, getting the biophysical information by doing your flaps, your flips, your flaps, your flims on your cell to get your biophysical information. And it's really that. We shouldn't lose sight of the bigger picture. It's not just about understanding the structure. It's understanding what else is it about that individual. And the more we understand that, the more relevance we'll have when we study it at all. So that's it. Okay? So hopefully it's entertained you. Hopefully if you weren't interested, you found the 10 football teams within your abstract. If you haven't and you get bored in anyone else's talk, don't, because there's a great lineup to come. Okay? And you can catch me tonight, and I'll point out the 10 teams if anyone's really interested. Okay. Actually, it was a real challenge for me. I had to go back to my notes to find out which they were. I knew I'd forget. So a very quick thank you to the team who really added a lot of data to these stuff that we have here today. The PIs whose data I've used extensively. Obviously, the Collington Lab has been fantastic in really helping us drive this forward. I am not a native electron microscopist. I am a light microscopist, which is why everything is... I always look on the bright side, huh? Everything is so positive. Okay. And the chemists, and also very kind thanks for these companies for trusting us to help work with them and develop it forward. And thank you for listening and putting up with that. Thank you.